Good day, students. This is a BCH 212 revision class in preparation for your examination. Now, these are very important areas that you need to pay specific attention to while going through your notes. You need to understand precisely the characteristics of cell membrane. Very, very important. Look at the characteristics of cell membrane. Membrane are structurally composed of lipid bilayer embedded with proteins along with carbohydrate on the membrane surfaces. Alright, so this is very, very important. Then another characteristic of cell membrane is that cell membranes are semi-permeable. They allow the transport of only a few specific ions and molecules. So the question is, what are the characteristics of cell membrane? Which number one, cell membrane is semi-permeable. Cell membrane are structurally composed of lipid bilayers embedded with protein along with carbohydrates on the membrane surfaces. And then you also say that cell membranes are amphipathic in nature. Amphipathic in nature meaning that what is on the other, one side of the membrane is different. That is the lipid bilayer. The outer leaflet of the lipid bilayer contain different phospholipid from the lipid layer in the inner leaflet. So you need to take note of that. So there are about, how many have I mentioned? 3. 1. Membranes are structurally composed of lipid bilayer. 2. Membranes are semi-permeable. 3. Membranes are amphipathic in nature. Okay, and then membrane contain hydrophilic head and then hydrophobic tail. Very important. That's 4. Membrane contain hydrophilic head, which is made up of the phosphate group, and then hydrophobic tail, which is made up of the fatty acid chain. So this is the major characteristics of membrane. Now that takes us to the very important aspect, which is the models of the membrane. The membrane models. So the membrane model, we have about 4 major membrane model. The lipid bilayer model. Now, what the question will be, they will say explain the membrane model with specific emphasis on the improvement of each model over the previous model. So that means when you are reading, you have to pay specific attention to the model and its improvement on the previous model. So the very first model was one called the Grandel model. It is known as the lipid bilayer model. This model simply says that the membrane consists of polar hydrophilic group positioned on the outside and the hydrophobic ends positioned on the inside of the membrane. So look at it here. Look at it here. So the hydrophilic head and the hydrophilic polar head, and then the hydrophobic tails. Okay, in the middle, and then you see another hydrophilic head in the inside. So water is on the surface and also on the inside. So that is how the first model, which is known as the lipid bilayer model, proposed by Overton and then Gotter and Grandel, 1925. Okay, so the next model is the sandwich model. Now, this one was proposed by Davison and Danieli. What did the sandwich model say? The sandwich model said that the membrane consists of 2 lipid layers sandwiched in between 2 continuous protein layers. 2 continuous protein layers. So look at it here. See it here. You see the lipid layer is sandwiched in between 2 continuous protein layers. The green up and down is the continuous protein layer. Then the inner yellow and red sandwich is the lipid layer. So this is what the sandwich model says, that the membrane is like lipid bilayer sandwiched in between 2 continuous protein layers. So the improvement of sandwich model over the lipid bilayer model is that lipid bilayer never gave account. They didn't say that they are receiving protein in the membrane. So in the lipid bilayer, there is no protein at all. But in the sandwich model, they say that yes, there is lipid, but the lipid is surrounded by a continuous layer of proteins. Okay, then that takes us to the next model, which is the unit membrane model. This one was suggested by Robertson. All right. So the model is saying that the unit membrane comprises of bimolecular lipid present in between outer and inner layer of proteins. Then in this model, the protein coating are asymmetric. Underline the word, the protein coating are asymmetric. So this is what distinguishes the unit membrane model from the sandwich model. In the unit membrane model, they are saying that the proteins, the continuous layer of protein that surrounds the lipid bilayer are asymmetric. In other words, the content of the outer protein layer is different from the content of the inner protein layer. So you can see what they are saying here. So this is the protein layer, the green one outside on top and the green one underneath. All right. So they are saying that this green one underneath is a contain different types of protein compared to the green one above the extracellular medium. So you see how it is arranged, and then you see the lipid bilayer sandwich in the middle. So see the difference. What is the difference between unit membrane model and sandwich membrane? The simple difference is that the unit membrane model says that the layer of continuous protein is asymmetric on both sides. So what is on the type of protein that is contained on the outer leaflet of the protein structure is different from the one in the inner layer. Then the next model is known as the fluid mosaic model, which is the fourth one. It's proposed by Singer and Nicholson in 1972. So this particular model proposed that the membrane is fluidic in nature with a mosaic structure consisting of phospholipid, cholesterol, protein, and carbohydrate. So basically, the difference between the unit membrane model and the fluid mosaic model is that whereas the unit membrane model is saying that the membrane is surrounded by a continuous layer of asymmetric protein outside and inside, the fluid mosaic model is saying that no, it is not the protein that is present on the membrane is not a continuous layer. Rather, it is scattered all around the surfaces of the membrane in a mosaic manner, and that the membrane is not just a rigid structure, rather it is fluidic in nature. Very, very important. See the model of the fluid mosaic model. You can see the proteins that are scattered. Look, this blue one that span from the surface down is the integral proteins or the transmembrane channel protein. Then we see the peripheral protein, those ones that are situated at the surface of the membrane, the blue circular structures. So they are called the peripheral protein or globular protein. So peripheral protein, globular protein. So you can see, then they said there are carbohydrates on it. See all this one that looks like a horn, and then the green glycolipids. All these ones are usually receptors and then sensitive structures for cell recognition and immunological function. So you see, this is the fluid mosaic membrane. You can see in the fluid mosaic membrane, the head is the yellow structure. That is the hydrophilic head, is the yellow structure, both on the surface and on the inside. And then you see in the middle, which is the tail, the hydrophilic tail that form the core of the bilayer. That is how the fluid mosaic model is arranged. This is another structure for you to appreciate too. So you should be able to draw this particular structure and label it accordingly. Always remember to put in the integral protein or the channel protein, the peripheral membrane proteins, the hydrophobic non-polar tails, then the hydrophilic polar heads, and then the phospholipid bilayer, and then the globular proteins, the glycoproteins, the carbohydrates, the glycolipids. Just include all this, and then the cholesterol. You see the green small structures there embedded inside the membrane core, the cholesterol. All right. So this is the membrane model, very important. So you should be able to explain. So what is the advantage of this? The advantage of this fluid mosaic model over other membrane. The advantage is that this is the model that best explains the structure of the membrane, and it is the one that is used the most widely accepted model of the membrane. With this, it explains why non-polar substances can actually cross the membrane. So, if the membrane is just the way that unit membrane proposed, that the membrane is surrounded by a continuous layer of protein, it then means that some non-polar substances may not be able to cross through the membrane. You see, but with this particular model, it has explained to us how that non-polar substances such as glucose, polar substances such as glucose can actually penetrate the membrane. So the unit membrane model did not give explanation as to how polar substances can actually cross the membrane, can actually move across the membrane. So in the fluid mosaic model, it was explained that polar substances such as sodium ion or glucose can actually penetrate the lipid layer of the membrane through the integral membrane proteins, and also by the mobility of the peripheral protein, which have the capacity to detach themselves from the membrane bilayer and bind to the polar substances, and embed the polar substances and carry them across through the lipid bilayer, and then transfer them into the intracellular fluid. So the fluid mosaic model is the model that can give an explanation of how polar substances can permeate the non-polar phospholipid bilayer of the membrane. Okay, let me say it again. The fluid mosaic model is able to explain how polar substances such as sodium ion, glucose can permeate through the non-polar hydrophobic membrane bilayer. And it explains that by saying that there are integral membrane proteins that serves as channel for the transport of those polar substances, and also that the peripheral protein have the capacity to detach themselves from the membrane core, the hydrophobic membrane, and bind to the polar substance, and then find their way again into the hydrophobic membrane layer and permeate it, and then opens into transfer the polar substance into the intracellular fluid. So the channels and the peripheral protein, the channel protein and the peripheral protein account for the reason why substances that are polar can permeate the non-polar membrane structure. And also the fluidic nature of the membrane is another important aspect of the fluid mosaic model, because if by saying that the membrane is fluidic in nature, it means that the membrane can gel and can also dissolve. So it's not a solid state. So depending on the temperature, the membrane can solidify and also the membrane can dissolve, just like oil. If an oil is exposed to a very cool temperature, it will gel. If it's exposed to high temperature, it will dissolve. So that is exactly what they mean by the gel sol nature of the membrane, and it is the fluid mosaic membrane that was able to give account of that. Okay, the unit membrane did not give account of, because the unit membrane presented the membrane as a solid unit. Okay, so that's very, very important for you. Then, having said that, we talk about the artificial membrane. What you need to know about the artificial membrane is that you must know the various types of artificial membrane: the phospholipid molecule, the flat phospholipid monolayer, the phospholipid bilayer, flat membrane type. See it here. The various types of membrane, artificial membrane. You see, you have the monolayer, you have the bilayer, then you have the black membrane, okay, which is the D. Then you have the E, which is the micelle, and then you have the F, which is the liposome. You should be able to explain them very well by drawing. Using diagram, you explain the various types of artificial membrane. So that takes us to the structure. So you can look at this. You can even give an example. You have dendrimer. Also, there are different types of artificial membrane: dendrimer, DQ actome, liposome, micelles, neosome. These are artificial membranes. Very, very important. And then you look at the what is called the polymer. Polymers are what is used in synthesizing artificial membrane. So also another important question that may be asked is for you to explain give examples of artificial membranes and their application. Examples of artificial membrane and their application. So simple and short, just tell them examples of artificial membrane: dendrimer, DQ actome, liposome, micelle, neosome. These are examples of artificial membrane. You can draw them. You can actually draw them. Just looking at the structure like this, you can draw them. Look at micelle, look at liposome. You can actually draw this structure to give examples of artificial membrane. You get your full mark. Then looking at these ones, they are what is called polymers of membrane, artificial membrane. So artificial membrane are formed from polymers. So polymers are a kind of substances that are formed due to the continuous addition of monomeric units. Example of polymer is cellulose acetate. It's a polymer. Why is it a polymer? Because it is made up of continuous chain of cellulose acetate. So this is the monomeric unit of cellulose acetate. The monomeric unit of cellulose acetate, look at it here. The monomeric unit. So which means cellulose acetate contains millions of this particular structure. Then what is the application? Let's go to the application of artificial membrane. The application is used in microfiltration. Artificial membranes are used in microfiltration. So what is microfiltration? You define it. It's a filtration technique that removes microorganisms and residual suspended particles through a membrane. They are usually used in the treatment of water. Another application of artificial membrane is ultrafiltration. These techniques suspend solids and solids of high molecular weight through a semi-permeable membrane based on forces like concentration gradient and pressure. Take notes. So very important, you need to know the applications of artificial membrane, and they are what are they? 1, microfiltration. 2, enzyme used in enzyme and organic acid purification. These are them. Then another important application is in ultrafiltration. You must know how to explain ultrafiltration, explain membrane distillation, explain reverse osmosis. So the reverse osmosis is simply used to remove ions and some larger particles from a water sample by using pressure. So the underlying force that is used in purification by reverse osmosis is pressure. Using pressure as a driving force. It's mainly used to treat wastewater. So you can see this structure here. This is explaining to you how reverse osmosis is done. Look at the semi-permeable membrane in the middle. This is the high contaminant water, highly contaminated water with high concentration of contaminants. See the pressure that is used to supply the water to push it through this membrane. So as the water passes through this semi-permeable membrane, the contaminants are trapped and the water passes, clean water will pass through the membrane, and then that is what is called reverse osmosis. Very, very important. So what is the force that separates that is used for purification in reverse osmosis? It is difference in pressure, use of pressure to force contaminated water through a membrane, and the membrane purifies, traps the contaminant and allows only the clean water to pass. Then we have membrane distillation. What is the force that is used in membrane that separates water and membrane distillation? It is the force of vapor difference across the two sides due to temperature difference. So this is heated. This one involves heat. Membrane distillation involves heating water, and then the difference in pressure due to the heat in the two sides is what will drive the purification process. Then we have the microfiltration. This one removes microorganisms and destroys them through a membrane. So then we have other application of synthetic membrane or artificial membrane, that they are used to purify enzyme. They are used to purify organic acids too. So thank you very much. So this is two major aspects of your examination. I wish you success in your exam. Thank you.